Hi my dear students, welcome to my biology class. Today let's begin our class with an activity. The name of the activity is the preparation of the temporary mound of human cheek cell. Activity preparation of temporary mound of human cheek cell. The materials required are Number 1, a glass slide. Two, a dropper. 3, an ice cream spoon or a toothpick. 4th one, a stain, methylene blue. Methylene blue stain. Then 5th one, a cover slip. Now let's familiarize each materials that are required for this activity. This is a glass slide. Here I use the ice cream spoon for doing this activity. This is the dropper with the methylene blue stain, the blue colored stain. When we apply stain on the specimen, it spreads over the specimen so that we can easily visible the parts of the specimen clearly when it is observed under the microscope. Next is a cover slip. You can see a cover slip like this. So it's a small, very transparent, thin cover slip. It puts over the specimen after the preparation of the specimen. Let's start the activity now. For this, we need to take the inner side of the cheek cell. So by using this ice cream spoon, you have to gently scrap the inner side of your cheek cell. So you get something like this with the saliva and some number of cells in it. So it is not visible with our naked eye. So we need to put 1 drop of methylene blue solution over this. For this, I take the methylene blue, then I put 1 drop of methylene blue solution over the specimen. The inner part of the cheek is made up of specific kind of epithelial cells, so that this specimen, the substance that we put on the slide, may contain a few cells of the epithelial tissue and a few saliva and other substances. Now I put 1 drop of methylene blue over this. Thereafter I put the cover slip gently over the specimen. Why should we gently put the cover slip over the specimen? When we put the cover slip suddenly over the specimen, what would happen? More air bubbles will be trapped inside the specimen. So when we observe the specimen under the microscope, we can see a lot of air bubbles other than the cell content. So for avoiding this, we have to gently put the cover slip over the specimen. Now the specimen is ready to observe under the microscope. This is the preparation of the temporary mound of human cheek cell. Did you get it? Now let's see how the cells look like when we observe it under the microscope. Now you can observe the image of the human cheek cells that are observed under the microscope. Now you look at the diagram, observe it carefully. You can see a darkly stained part which is present at the center of the cell. The dark spot is called the nucleus. Around the nucleus there is cytoplasm and there is a cell boundary, that is cell membrane or plasma membrane. These are the 3 functional parts of the cell. We have already studied about one of the functional segments of the cell, that is the cell membrane. We have learned the mechanism, its features, its functions, etc. in the previous class. Today we are going to learn more about another important part of the cell, that is the nucleus. So now we are going to learn more about nucleus. So today's topic is nucleus. So what did you observe there in the human cheek cell? In the human cheek cell, the nucleus is located at the center of the cell. This is the nucleus. In animal cell, the position of the nucleus is at the center. We can do the same experiment with an onion cell. So peel off the onion and put the peel over the slide, then put one drop of methylene blue stain over it, then put a cover slip and observe it under the microscope. So the cell can be observed like this. So this is a plant cell. Human cheek cell is an example of animal cell. In the animal cell, the nucleus is located at the center of the cell, while in the plant cell, the nucleus is located at the periphery of the cell. Suppose this is a plant cell. You know that plant cell has a cell wall and a cell membrane. Cell wall, then cell membrane. Then it has a large vacuole in it along with the cell organelles. So the nucleus is located at the periphery of the cell. Why? Because it has to accommodate large vacuole in it, so that it is slightly near the periphery of the cell. Did you get the difference? The position of the nucleus both in animal cell and the plant cell. Do you remember the name of the scientist who discovered nucleus? That is Robert Brown in 1831. Now let's study the structure of nucleus in detail. Now let's study the typical structure of a nucleus. A nucleus is spherical in appearance. So here I draw the spherical structure of the nucleus. So you look at the board. You will understand how to draw a spherical structure of nucleus. Okay, so spherical structure of nucleus. The outer covering of the nucleus is called nuclear membrane. Nuclear membrane. The nuclear membrane is double layered structure. It is not continuous. It is interrupted at specific spots. Those are known as nuclear pores. Nuclear pore. Okay, the nucleus consists of a jelly-like substance called nucleoplasm. Nucleoplasm. In the nucleoplasm, there is another spherical structure called nucleolus. Nucleolus. And another important part of the nucleoplasm is the chromatin network or chromatin thread. Okay, this is the chromatin thread or chromatin network. These are the important parts of a typical nucleus. Then let's study the structure and function of each part of the nucleus in detail. First of all, nuclear membrane. Okay, nuclear membrane is the outermost covering of the nucleus, which separates the content of the nucleus from the rest of the cell. That means it separates the cytoplasm from the nucleoplasm. Suppose this board is a cell. So the boundary is the cell membrane, and this part is the cytoplasm, and this is the nuclear membrane, and inside the nucleus there is nucleoplasm. So the nuclear membrane separates the nucleoplasm from the cytoplasm. It is a double membranous layer. Double layer. Okay, did you get it? The next one is nuclear pore. This is the nuclear pore. The nuclear membrane is not continuous. It is interrupted at specific intervals. Those are known as nuclear pores, which allow the transport of molecules across the membrane. That means in and out of the nucleus. That means some substances get into the nucleoplasm for certain functions or activities of the cell. Some other components move from the nucleoplasm to the cytoplasm. So this is carried out with the help of the nuclear pores that are present in the nuclear membrane. The next part is nucleoplasm or nuclear sap. Nucleoplasm is also known as nuclear sap. This nucleoplasm is the site of cellular activities. That means the cell division is controlled by the genetic material that is present in the chromatin thread or chromatin network. Then it provides a site of cellular activities, especially for cell division and protein synthesis. That's about nucleoplasm. It is a semi-fluid part of the nucleus. Here you can observe a darkly stained part, that is the nucleolus. It involves mainly in the production of proteins in the cell. It is storehouse of rRNA. rRNA means a ribosomal RNA. Okay, then nuclear membrane, nuclear pore, then nucleoplasm, then nucleolus. Next, let's study the most important part of the nucleus, that is chromatin thread or chromatin network. The chromatin thread or chromatin network is actually the genetic material of the organism. It could be either DNA or RNA. The chromatin network is actually the genetic material. Which are the two major genetic material? RNA or DNA. So what is DNA? It is a nucleic acid. It is deoxyribonucleic acid. DNA is deoxyribonucleic acid. The DNA is the genetic material in most of the living organisms, including human beings. RNA is the ribonucleic acid, which is the genetic material in some living organisms, especially the viruses. Now let's study in detail about the structure of the DNA. You might have seen the 2D structure of DNA like this. Have you seen this? The two strands with the that means a ladder with the steps like this. This is the 2D figure of the deoxyribonucleic acid or DNA, and it is composed of chemical substances, chemical compounds like sugar, phosphate and nitrogen compounds. Then what is the role of this DNA or RNA? The genetic material stores the information for producing various proteins. Those proteins help in the expression of various traits or characters in the living organism. So the all information about a living organism is coded in the DNA or RNA in the form of chemical compounds, so that DNA and RNA are known as genetic material. So I am like this, that human beings are like this because of the information that is coded in the DNA. The DNA contains all the information about the body features of a human being, so that the body features are appeared like this with the help of the genes present in the DNA. Actually, the chromatin thread is made up of not only DNA but also with the protein. So we can give an equation like this: chromatin material is equal to DNA with the protein. So here we are talking about the DNA. DNA has functional segments and non-functional segments. The functional segments of DNA are known as genes. Functional segments of DNA are known as genes. So what these genes actually perform? They carry the genetic information for controlling a specific trait or characteristic in living organism. So genes are known as the unit of inheritance. Unit of inheritance. That means the organisms or young ones receive the hereditary characteristics from their parents through the genes only. So that these are known as the unit of inheritance. The genetic material is observed as a thread-like or a network inside the nucleus when the cell is at the resting phase. So this is the structure of the nucleus at this time of the resting phase of the cell. When the cell is about to divide, that means when the cell undergoes cell division, this chromatin thread or chromatin network condenses to form chromosomes. So actually the chromatin is made up of DNA and protein, and this DNA and protein condenses to form chromosomes at the time of cell division. So look at the diagram. Suppose this is a chromosome. Chromosome is a rod-like structure. Chromosome is a rod-like structure. This genetic material is condensed to form what? A chromosome. So high-level packaging or condensation take place during the cell division. This thread-like DNA is very lengthy in case of complex living organisms. In the case of human beings, it is 2 meters inside the nucleus. So the lengthy DNA undergoes condensation to form the rod-shaped structure called chromosome. This is the diagrammatic representation of a chromosome with the condensed genetic material inside it. That means chromosomes carry the genes. Genes are the unit of inheritance. We all know that our body has a controlling center, that is our brain. Brain controls and coordinates all the activities of the body. Similarly, nucleus is the controlling part of the cell. Then it controls all the cellular activities, including the cellular reproduction. Cellular reproduction means here cell division. One cell divides into 2, 2 into 4, 4 into 8. Like that, the cell divides exponentially during the cell division. So it is also under the control of nucleus as it carries the genetic material, either DNA or RNA. Not only during the cell division, but also it plays a major role in protein synthesis in living organisms' body, because different proteins are required for the living organisms for the expression of their characteristics, for the production of hormones, for the enzymes, for the expression of a trait like our hair color, like that needs a different types of proteins. So new proteins are synthesized in the cell with the help of the nuclear material. Now, did you get the functions of nucleus? The structure and functions? All cells in the living organisms do not have this typical nuclear membrane. So, some cells like prokaryotic cells. Prokaryotic cells are primitive cells, prokaryotes. Pro means primitive, karyon means nucleus. Primitive nucleus. The organisms like bacteria, blue-green algae, they do not have a typical nucleus like this. They lack a nuclear envelope or a nuclear membrane like this. So their genetic material is simply located at the center of the cell. So such cells are known as prokaryotic cell. While other living organism cells have a typical nucleus with a nuclear membrane or nuclear envelope, those cells are known as eukaryotic cell. Now you can observe the image of a prokaryotic cell. It lacks most of the cytoplasmic cell organelles or cell structures. Many functions of the cell organelles are performed by poorly organized parts of the cytoplasm. The chlorophyll in photosynthetic bacteria is associated with the membranous vesicles-like structures, but not with the plastids as eukaryotic cells. That means plastids are well-developed structures for performing photosynthesis. But these type of structures are absent in the prokaryotic bacteria, photosynthetic prokaryotic bacteria. Photosynthetic prokaryotic bacteria means the bacteria is able to do photosynthesis with the help of the pigment chlorophyll, but they do not have a clearly developed structures, plastids like a eukaryotic cell. Next, you can observe the image of a eukaryotic cell. A eukaryotic cell has a typical nucleus with cell structures in the cytoplasm. Each cell structure or cell organelle has a specific function in the eukaryotic cell. Now, let's study the difference between prokaryotic cell and a eukaryotic cell. Prokaryotic cell and a eukaryotic cell. Pro means primitive. Primitive. Karyon means nucleus. That means these type of cells do not have a typical nucleus with a nuclear envelope or nuclear membrane. Those type of cells are found in the living organisms, especially the microorganisms like bacteria, blue-green algae, etc. Blue-green algae, etc. Okay, that means their genetic material is centrally located in the cell without the nuclear envelope or nuclear membrane. The genetic material is very simple. It will not be more than one chromosome, so a single chromosome is present in such cases. Eukaryotic cell means a typical nucleus is present in such type of cells. That means a nucleus with a nuclear membrane. Nucleus with a nuclear membrane are found in the eukaryotic cell. This type of that nucleus, typical nucleus, is found in the living organisms like plants, animals, including human beings. Example: plants, animals, including humans. The organisms having prokaryotic cells are known as prokaryotes, while the organisms having eukaryotic cells are known as eukaryotes. Now let's understand more differences between the prokaryotic cell and the eukaryotic cell. The size of prokaryotic cell is generally small, that is about 1 to 10 micrometer. 1 micrometer is equal to 10 raised to minus 6 meter. While the size of the eukaryotic cell is generally large, it is about 5 to 100 micrometer. The next difference is about nuclear region. The nuclear region of the prokaryotic cell is known as nucleoid. It has no nuclear envelope or nuclear membrane. While the eukaryotic cell has a typical nucleus with a nuclear envelope or a nuclear membrane. The prokaryotic cell will have a single chromosome, while the eukaryotic cells will possess more than one chromosome. Prokaryotic cells do not have membrane-bound cell organelles, while the eukaryotic cells have membrane-bound cell organelles, which are highly developed also. So, what's the meaning of the cell organelles? The cell structures, the structures that are present in the cytoplasm. The prokaryotic cells do not have such well-developed cell organelles. They have poorly developed cell organelles, while the eukaryotic cells have highly developed cell organelles or cell structures. Those cell structures are found in the cytoplasm. Each cell organelle or cell structure has a typical function. We will learn about cell organelles in the upcoming classes. I hope you understood the topic well, the structure and function of the nucleus, the differences between the prokaryotic cell and the eukaryotic cell. With this, I conclude my session. Then thank you and have nice moments.